Take the microphone here because I can't stand still usually. I like to walk around. Thanks for coming this morning. And like most others presenting, there's a whole suite of people that are actually involved in doing all this work, and this isn't even the full list of people. But what I'll talk about for the next 12 minutes before I get the 3-minute warning on this side over here is the cultural transformation of the boreal landscape over the past couple millennia. And for obviously all of you sitting here, the idea of human impacts on the landscape is nothing novel. So there's nothing new that I'll tell you in that regard. But what I think the problem that we're facing is in the limnology and ecology community is they've kind of forgotten the lessons that have been learned over the past 30, 40 years in terms of environmental archives. And this is certainly true, I think, in the Swedish community. So here I show you a picture of 2 lakes. One is called Lusevatten, a classic clear water, acid-sensitive lake in the southwest of Sweden, and a lake called Dragsjön up in central Sweden, I guess you could say a classic boreal humic lake. Looking at the pictures, what human impact do you have? Beautiful, pristine landscape, old forests that have grown 100, 200 years of age. There's no human impact. So if you talk to a limnologist or an ecologist and you're interested in, say, the effects of climate change, you're starting from a natural system. So if you go to a limnology meeting, and even when I went to Acid Rain 2 years ago, I mean, there's a community that worked with limnology very actively for 30 years ago. Wait a second, that was Swinglish, 30 years ago. My English has really gone down over 20 years in Sweden. This is their idea of long-term. And in fact, when I went to Acid Rain meeting 2 years ago, this was probably the longest term that was meant there. So you'll have people talking about long-term being a 3-year study, a 5-year study, or in the best case situation, a 30-year study. So here we have lake water TOC, and it's from these 2 lakes here, which are part of the national monitoring system in Sweden. So every little dot here is a national monitoring lake, for which there's typically about 30 years worth of data. So one is a humic lake, Djupsjön, and then one is a classic clear water lake, Lilla Örsjön, in the southwest. And like many lakes in Sweden, lake water TOC has been increasing over the past 30 years. And so for a limnologist, this is it. This is their perspective on the carbon cycle and how it's changing. And this pattern is being observed across a lot of northern systems in Scandinavia, the UK, and then even in the northeastern part of North America, where every red and orange dot is a lake where lake water TOC has been increasing. And of course, long-term in this context is 14 years. And then there's a lot of discussion about what are the impacts of carbon cycling, of this brownification, as it's called. It'll change the metabolic processes in lakes. It'll change, obviously, clarity of the lakes. It'll change the energy dynamics. And even with the change in the quality of the organic matter, it could also affect mercury cycling. So a lot of these danger scenarios that are portrayed. And the 2 leading hypotheses for this are, 1, it's simply a recovery from acidification. As you reduce the input of mineral acids, you're restoring the function of natural organic acids. And there's lake water pH and how it's increased over the past 30 years. The other potential explanation is that it's climate change, that with increasing temperatures, you're increasing the transfer of labile organic carbon from soils to lakes. And so these are the 2, I guess you could say, competing hypotheses. So if you're going to project what's happening in the future, the question is this simply a restoration of lake water TOC and natural acids? So the idea is then, what is the natural baseline? So where is recovery? Or 2, is this an unprecedented increase, which is a function of climate, and in some cases there's some discussion of modern forestry practices as well. Which would mean that this baseline was somewhere back here. So this is actually borrowed from work that Rick Batterby had summarized back in 1999. So again, this is, for us in this community, this is nothing new. So is this a combination of stresses, but this is our sole viewpoint? And then the question is, which we know again from our community, is there also something that's underlying those changes? And to sort of backtrack, these are things that we know. So if we go to the Scandinavian landscape, we've gone from a glacial landscape to this landscape that we have today. We know all about these things concerning natural landscape development, forest succession. In Sweden, we had spruce immigration, and of course, natural climate changes. And we know that human impacts have been superimposed on that system for hundreds, if not thousands of years. I mean, those of you working in the Alps or the Pyrenees know that this is several thousand years of human impacts. And again, going back to work from Ingemar Renberg, 1990. So, I mean, this is 20-year-old data. This was the perspective already back then. So here's the monitoring data from Lilla Öresjön up until 2015. And then this was the high-resolution pH record that he had done back then. So again, this is nothing new. The limnologists knew this 20 years ago. So you have the deglaciation, landscape development, and soil development. You have forest succession and a gradual natural acidification of many of these ecosystems in the north. And then this was interrupted by diverse land use practices over the past 1,000 to 2,000 years. So again, this is old news. We know this. And we knew this a while ago. So we have this dimension then of human impacts. Landscape utilization I would prefer to use rather than land use. So this is not new. So here we are back in the day of brownification, and we're back to this time interval. Monitoring started in 1985, and we're up here to the present. This is the perspective that most limnologists and ecologists are operating on. But we knew this. So how does this relate then to carbon cycling? So one, carbon cycles are long. So if we go to this lake Dragsjön, if you just simply take the tree species in the catchment, what are the timescales of carbon cycling? I mean, these trees are 100 to 200 years old. If we go to the forest soils, Jonathan Clementer, sitting further in the back, has dated the humus in these types of soils, and it's 150 to 500 years old. And in extreme cases, if it gets a chance to build up in the absence of fire, it can be 5,000 years old. And many of these lakes are connected to peatlands, and these can be 8,000 or 10,000 years old. So carbon cycles operate over very long timescales. It's not a 30-year cycle. So a tool that our group has developed over the past 10 plus years, originally by Peter Rosén back in 2005, and then currently it's Karsten Meyer-Jakob who's taking the main responsibility for this, developed a transfer function based on the spectral qualities of surface sediments which were calibrated against lake water carbon so that we can reconstruct lake water TOC. And the calibration set includes lakes in Sweden, about 100, 20 lakes in Finland, over 100 lakes in Canada from John Small as well, and then we can compare the reconstructions against the monitoring data. So here's 2 Swedish lakes. In blue are monitoring data, and then different iterations of the model for lake water TOC, and then even tested it on a UK lake, which is outside of the calibration set. So we have a pretty good ability to reconstruct at least the past 30 years. So if we jump to the Holocene, right to it. So we have 18 lakes where we have geochemistry, we have this lake water TOC reconstruction, biogenic silica, pollen, and diatoms. But I'm just going to show you a cluster of 3 lakes, and they're all from this area here in central Sweden. And essentially all 18 lakes look quite similar to this. Early in the Holocene, you have an increase in lake water TOC, and more or less not much happens until about 1,000 years ago. Some small changes, and I'll go through those in a moment. But then here, over the last 500 to 1,000 years, we certainly have an unprecedented decline in lake water TOC compared to any other time in the Holocene, if we exclude the post-glacial period. So if we look at what are the possible factors behind this, if we look at climate, we know climate has changed quite a bit over the Holocene. Again, ignoring the initial post-glacial period, this is a pollen reconstruction, which is typical for basically any lake that's been done in Sweden. Vuoskojärvi of Bjuna et al. There's a whole collection of lakes in Norway that they've reconstructed for. Heike Seppe, I don't know if you're here, has done a number of those in Sweden as well, and summarized for all of Northern Europe, and they all look quite similar. You have elevated temperatures during the Holocene thermal maximum, but not so dramatic changes throughout the rest of the Holocene. But the lake water TOC, there's nothing happening in conjunction with temperature. So we have fairly large changes in temperature, but not fairly large changes in lake water TOC. A second very major impact in Sweden then is the immigration of spruce. And here, 2,500 years ago, you see pollen records for all 3 of these lakes. Here comes spruce, and no impact on lake water TOC. But again, we have this unprecedented decline here. But what we do know, and again, this is not a novelty for those of you working in the Pyrenees or the Alps or other mountain parts of mainland Europe, I'll call it, the landscape, even in the boreal forest, was extensively used. And this particular mire, in Swedish it's called fäbodmyran, which is summer pasture farm mire. And haymaking was done in these things from the Middle Ages up until only 100 years ago. The hay produced on the mires determined how many animals you could have on the home farm for the winter. And then, so in the summer when they were out here cutting the hay, they were grazing the animals out in the forest. So sheep, cows, and goats. And goats are really effective for chewing up the forest and opening it up. So for these 3 lakes that I'm showing you, here's 2 of the lakes, Dragsjön, Långälvsjön, and Trisjön. And here you can just see a 1 kilometer scale. Every one of these red circles represents one of these enclosures for forest summer farms. And this was an enclosure where a village, so there could be 3, 5 to 10 farmers who would bring their animals here, and at night they were brought into the enclosures, and during the day they'd be grazed out in the forest. No lake is more than 2 or 3 kilometers from one of these farms. There's not much known about them. The first census wasn't until the late 1600s, and almost every one of these existed in the late 1600s, is documented. But there is references in some of the few texts that are available that these were certainly in place, in some cases, already back in the 1300s. And so this was going on from each one of these locations. Yet you have this beautiful lake today. It's considered a nature reserve, a wilderness reserve actually. And you had these types of activities. So when these activities really began to expand, at the top I have total arboreal pollen, and you see a decline in the forest canopy represented by the total arboreal pollen. And then the next values here are for cereals. They would be cultivating cereals in some of these low-lying areas around the enclosures, but of course in the forest you're not going to be growing very much. But where they had some soils, they would plant grains. And so we have opening of the forest, we have some cultivation, and that's coinciding with the declines in lake water TOC. But then as an added dimension, because we always have the problem that pollen is diffuse, is that all those activities representing what's happening 1 kilometer away, 5 kilometers away. So what we added is coprastanol. It's a fecal biomarker. And if you're measuring shit in the sediment, then the animals are shitting around the lake. Sorry, but it's just the way it is. So if this is getting there, this isn't an indirect proxy. This means the animals are there at the lake. They are grazing around that lake. And we see this already back around 600 years ago, maybe even 600 AD, maybe already back around 300 AD. So that's pretty hard evidence that they're grazing already at that point in time. And then it accelerates in the 1800s, which is known from historical texts. So we have a very good coupling between this landscape utilization, which was using essentially the whole landscape. There wasn't anything that was not being used. And this decline in lake water TOC, which we see essentially in all 18 of the records that we've worked on in different parts of central and southern Sweden. So for the limnologists and ecologists working with brownification, they need to take into account more than just the question of, is this a restoration of the function of natural acidity with the reduction of acid rain? They need to take into account more than the fact that this could be a climate change component, that there's actually a historical dimension to it that's underlying it. This wasn't a steady state before temperatures were increasing over the last 100, 150 years, depending on how you want to define that. That there's actually an underlying dimension to it, which we know from acidification. So with that, I also want to introduce that in 2018, the Paleolimnology Symposium will be combined between the Paleolimnology Association and the Limnogeology Association in Stockholm, 18 to 21 June 2018. So we can continue the discussion at that time. And Malin Kylander is sitting in the back, who's also one of the organizers of that. Dragged you into Malin. So thank you very much.
Was there native grazers before the stock were introduced, and are you seeing any impact from that?
From?
Native grazers.
Mm-mm. No, I don't think you didn't have that concentration of animal density, I guess you could say. And with the animal grazers, I mean, the thing that they write about historically is already as you come into the 1700s when forest resources were becoming more marginal because of the need for wood for metallurgy, is animals like goats were banned because of the damage they would do to the forest. So there's an intensity that you didn't have with the native grazers.
I'm going to talk today about some work that has been published last year, 2016. It was mainly a work that was made by Jean-Philippe Génie, who used to be a postdoc in my lab. But as you can see, there was a lot of contributors, and you will realize from different places in North America and Europe, and you will see that it's because we compiled a lot of data, and it was a lot of effort to arrive to the conclusion I will try to convince you with. It was also some work that was triggered by the former VARV working group of pages, and this is also a good illustration how pages can help you to put everything together. Oh, this one, okay. No. Which one is it? Okay. So when we look at lakes, as the previous speaker already talked about, we can think that the lake is forced by local and global forcing. At global forcing, we will think that temperature will be one of the main driver on lake oxygen content. Temperature, higher temperature, you decrease the amount of dissolved oxygen in the water, and you can also increase the thermal stratification. Local forcing that might be made by presence of cities or agriculture that increase the amount of nutrients in the lake, and more nutrients means more primary production, as you know, and more primary production is also a potential candidate for increasing the hypoxia in lakes. If we look at the marine hypoxia over the world, there was this map that was provided several years ago already, and you can see here the spreading of these dead zone, these red dots that are on the map here on the background. This is the size of the location and the size of these dead zone, and you can see that they are all located on coastal zones, and they are also located where they had a lot of high density of population and high economical activity. If you look at these dead zones, and this is based on monitoring data, it's really starting in 75, this increase of the number of these dead zones. Here you have just across the time here, the number of coastal sites that are recording hypoxia. So what's happening globally in the lake? And the problem in the lake that there are very few long-term monitoring data, as Richard illustrated. There are a few examples in Central Europe, like Lac Genève and other lakes, but there is not a lot. So we turned to the sediments, and our specialty, you say that was the initiative for the VARV working group. So what is nice about the VARV is that if you have benthic activity, you will have all the sediment mixed. And if you have no benthic activity, you will have, if the conditions are okay for forming them, you have preservation of these laminations. What is also nice is that these VARV, they are basically found everywhere in different kind of settings. So you can basically compare different watersheds between them. Here, this is an example from Lac du Bourget, within the same lake. This is a shallow core where there is a lot of oxygen in the water column, and this is a deep core over there where it's in the deep side where there is no oxygen in the water column. So, well, how a VARV here is just an example. You can have up to 4 layers: a spring deposit, a summer deposit, fall and winter. So this can be very well recognized and defined. In some places it's more complicated, but this is just an example of how it works. So what we started to do is to make a compilation of all in the literature of all the existing valve record. And you have two kinds of record. You have lakes that are naturally valved, so there is hypoxia naturally. And then you have also lakes where you only have the appearance of hypoxia only in the upper sections of the sediment. One of the good things is that if these layers are annual, you can count the layers, and you can also have a first record of the appearance of the hypoxic conditions. So here is the result of our compilation that we published in Global Change Biology. In this world map here, the red dots are the lakes with this recent hypoxia, and the green are all the lakes that are with natural hypoxia. So these lakes are naturally valved. Now, if we plot the number of lakes recording hypoxia against time, you see here is the red curve. You see that it is starting really way earlier compared to the marine hypoxia. And so basically, it started to spread after 1900, 70 years prior the coastal zone, and definitely before the rise of temperature and any significant change in climate. So one of interpretation of this early occurrence of hypoxia is that lakes are way closer to the source of the pollution. We wanted to have a little bit more information about the reasons of this occurrence of new hypoxia, and we turned to Europe. We focused on Europe because we know in Europe there are a lot more reconstructions. And in Europe we still have 51 lakes that record recent hypoxia, almost 100 lakes with, that are naturally valved. And we also looked at another set of lakes that were our benchmark, so to see if we have a coverage that is representative of the diversity of different watersheds, elevation, latitude, and so forth. So for each of these watershed, we made a GIS looking at the perimeter, the area, the mean altitude, the watershed temperatures, and precipitation. And we look to also the human parameters, the amount of cultivated area, the forest, the grass area, urban area, gross domestic product, human footprint, which is an indices that have been created by the WWF, and population density. So we have all this information for today, and for some of these parameters there are some historical data. And for instance, here from Europe, we have a time series. We could extract from these atlases here that has been created by these people. We can extract a time series of land cover that we extract and we include in our GIS. So that's an example. So first we wanted to see if we have a change indeed in the land cover in the watersheds that we have selected. So you have here our sampling, the watershed that we sampled. And here we have the changes in the cultivated area. We did a non-parametric Mankandal test to see if there was a significant change in the surface area. And all these dots here along this scale, if it's a positive score, that means that there is indeed an increase in the surface area of cultivated land. So that's quite significant here for the cultivated area, and same thing for urban area. A little bit, you can see there are differences. Let's say in northern Scandinavia, there is the increase in the urban area is not that important. Anyway, so let's go back to the original data set. In all these watersheds, we calculated that's the curve I already showed you here. That's the amount of lake that are recording hypoxia here. You can see the increase in the early 90s, and you see at the top here you have a plateau. So that means that in the 80s, we stopped basically the increase of hypoxia, and it's probably due to the efforts of the European countries to stop pollution and input of nutrients that are phosphorus-based. If we compare to the temperature, you see that hypoxia, a temperature reconstructed from all the watersheds we have here, you see that increased temperature is way after the increase of the hypoxia. So temperature cannot be the reason for this increase. Precipitation, you see, on average, we have no changes. Now, if we look at the change in the urban area, in all the watersheds, you see that that's the number of lakes with hypoxia here again. And here you see the increase of surface area in the watersheds in the lakes that developed new hypoxia. And you can see the two curves are really parallel. Then we looked at the cultivated areas. So the previous slides here I showed. And you see that the amount of cultivated area started way earlier compared to the VARV record here. One thing that is interesting is that in the cultivated area, here's the time where phosphorus and nitrogen-rich fertilizer were introduced into the cultivation practices. So a last slide here with a lot of information. You have, for each of these diagrams, you have 4 curves. The red curves is the one of the watersheds that have developed recent varves in the sediments. Then you have the yellow, this orange curve are the lake that with natural varves, and the 2 other curves are our benchmark lakes that from the lakes that we wanted to compare with. And as you can see, the only factor that we found that was really different compared to the benchmark lakes are the lakes are the watershed that have a significant increase in urban area in the watershed. So our conclusion is that some few conclusions here, that the spread of hypoxia in lakes started in the 90s, in the early 90s. Temperature is not responsible of the onset of the hypoxic condition in lakes, and that is global in Europe. The spread of hypoxia in Europe is best explained by the expansion of cities, and there is, for the moment, no signs of recovery of bottom water oxygenation. And this is bottom water oxygenation because there are some program where you are successful in oxygenating the epidemium, but it's not affecting the bottom waters. And one of the reasons that is probably phosphorus is recycled constantly in the lake. It takes a long time to be stored in the sediment. It's also, I think, a good message to send to the developing countries. You should take care of your lakes right away, not wait that there is something happening, because it's very difficult to recover from this situation. And with that, I will thank you for your attention. Yes, that's a good question. If you read the paper, we specifically mentioned that we have a selection criteria that only includes lakes with a certain bathymetry and where we are sure there is no huge fluctuation of the lake level.
Hello, I'm going to present some of the results of a recent project about the evaluation and monitoring of global change in 3 high mountain lakes. Okay. We have in Spain, we have 15 national parks, and some of them have beautiful lakes and wetlands, as you can see in this slide. We are working in these two, in Picos de Europa National Park and Ordesa and Monte Perdido National Park. Well, as in the previous presentations have said, the main threats of these lakes are the eutrophication, pollution, tourism, and global warming. And the main challenges are to have an adequate management. The Enol Lake is located in a beautiful landscape, as you can see here, of green meadows and Atlantic forest in an area. It's here, it's in Asturias, in a very complicated tectonic and geological area, surrounded by limestone formations. Here you can find all kinds of touristic activities in the lake, close to the lake. You can go to the Puerta Ciclista Espana. You can also go to celebrate the Shepherd's Day. You can get into a traffic jam in summer. And in addition, you can find a lot of cows, a lot of, the population of livestock population is very high. We also have some additional problems. They have a kind of a strange practice with the vegetation, and they used to clear cut areas of shrubs, like here, in a steep slope, areas like the one is marked here with the blue arrow. And then this is causing a this has negative consequences in the limnology, because it's increasing the erosion and the organic matter and clastic sediment inputs to the lake. We also have a mine, an iron and manganese mine, very close to the lake, and the important consequence is that they modify the hydrological regimen of the watershed. They built, this is an image of the lake before the construction of the road, and when they constructed the road, it's here, here, they increased the lake level, so they made like a dam here, and the importance of this flooding is that they flood this area from here, a small platform, green meadows with a lot of organic matter. The construction of the road also had important consequences because now we can find a lot of rills, gullies, a lot of erosional features all around the lake, like you can see here. Then in this project we have been doing a lot of analysis. I'm going to show you only some of the results of the multi-proxy study of the sediment course. Okay, I'm going to go. We wanted to know the basic limnology that was unknown so far, and we wanted to know by means of the study of the sediment record and the soil, we wanted to know when and why the lake started to be so anoxic that is now, and what is the influence of mining or other human activities in the environment. Yes, the final issue was to define a global change monitoring program. The lake, I'm not going to show the results of the limnology, but just only to say that the lake has a, there is a development of an increasing hypolignetic anoxia from July to November. The thick layer of anoxia can be up to 9 meters in July or August. And we know now that there is, the phosphorus values and the phytoplankton biomass is very low. So we think that there is a lot of allochthonous organic matter going into the lake. In the soil study, we have we know now that the soil is very carbon rich. If we take a look to a vertical profile of the soil, just here we see that the upper horizons of the soils are very rich in organic matter. And if we take a look to the profile, the organic matter composition of the soils, we can see that the areas close to the lake are very rich in organic matter. These areas are also very rich in people, tourists, and cows. So they are removing all the horizons of the soil. There are at least 3 previous papers that deal with the sedimentary record of the lake canal. Oh, pardon me. Okay. We took several sediment cores in a trunk set from the deepest area of the lake to the shore here, and we correlate our results with the previous papers. There is a good, perfect correlation of the total organic carbon profiles with the previous one, and we have used the H-depth model of Lopez Merino et al. With the correlation of the total organic carbon. Just a quick look to the kind of sediments that we can find. The 50 upper centimeters of the core located at the deepest part of the basin is made of black to brown sills. We don't have lamination of barbs in these 2 units. As you can see here, there are 2 units. A lower one with calcium poor. All the core is calcium poor, but this area from here. In this lower unit, we have an increase in the bromium content. In the upper unit, we have an increase in the clastic content, and in the 5 to 4 upper centimeters, we have an increase in the siliciclastic content and the bromium content. In this core, there is a perfect correlation and match between the bromium as measured by X-ray core scanner and the total organic carbon. In the 3 other in the 2 other cores located at 12 and 4 meters depth, we can also see this increase in the total organic carbon in the upper part of the core. If we plot the carbon and nitrogen ratio versus the delta 13 carbon, we can see, as we can expect, that the cores located at the deepest part of the area are close to the Lacustrine-Alger value, and the cores located far away from the deepest part of the area are more influenced by the land plant values. If we plot, I'm not going to go into detail, but if we plot these values, the type of organic matter, the total carbon nitrogen ratios, delta 13 carbon and delta 15 nitrogens, we can see also the same kind of data. There is an increase, a clear increase in the upper part of the core. Even the highest values are from the top of the core, corresponding to the 17th, 18th century, the Little Age. In order to explain and to complete this information, we made a biomarker study. We analyzed the kind of N-alkanes and other organic compounds in the sediments, and we know now that the paper is already published in Organic Geochemistry. We see that there is a predominance of N-alkane C31 in almost all the cores, but we have increase, or we have more algan. We have a contribution of the C17 N-alkane that is corresponding to an input or alkane components. Okay, I'm going to go to this. What we saw also is a pollution, a permanent pollution from the 17th century of organic compounds coming from activity of animals, because we found this organic compound along all the cores, from the bottom to the top of the core. If we take a quick look to the trace metals, the most significant trace metals, we can see also that there are 2 areas. This is the H, it's not depth, it's just, okay, well, we can see 2 different areas with a strong influence of mining. There is an older one that you can see a zinc and lead increase, and there is another one here that it's more or less coincident with the activity of the mine working in the area, that we can also see a lead and zinc, chromium, and a slight manganese increase. In the upper part, we can see it's increase in arsenic, copper, even manganese, some phosphorus, and talc that can correspond to atmospheric pollution, both from automobiles, industry, or who knows. Okay, and last year, in collaboration with Jorge, that is over there, we started to monitor the atmospheric deposition on the lake, and we have not very nice surprise because the rain, the chemical composition of the rain is full of zinc, lead, and copper in this beautiful national park. We also are studying the filters, the dry deposition, and we can see also small particles with lead, zinc, and manganese. Just as quick, fast conclusions of this study, we can say that there is a strong human impact in the area. Livestock has contributed in a negative way in the evolution of the lake. There have been changes in the hydrology due to the construction to the access to the road mine, and the tourism and clear-cut vegetation practice are also increasing erosion. In addition, we have atmospheric pollution. Yeah, we have atmospheric pollution because of the regional and local mining, industry, and automobile traffic. So the input of alloctonous organic matter, the apparent increase in bioproductivity, and the upper part of the 5 upper centimeters from the 70s, and increasing temperature is causing an intense, we think that it's causing an intense hypolymnetic anoxia that may be very negative, that might have very negative consequences on the ecosystem in the future. Thank you.
Thank you. You said you saw an increase in bioproductivity in your lake, but you said also that the phosphorus concentration were below 6 micrograms phosphorus per liters.
Pardon me?
You say that the phosphorus concentration or phosphate concentration were below 6 micrograms phosphorus per liters in the water column.
Yeah.
But how do you explain an increase in primary productivity at so low phosphorus levels?
We think that the increasement in bioproductivity, we deduce the increase of the bioproductivity because of the biomarkers of the sediment. But when we took the samples to analyze phosphorus, the phosphorus value was very, very low. I understand your question.
Do you see some development of biofilm around the lake? So it cannot be it. Okay. Okay, thank you.
I'm very happy to be here and get to present some of the work I've been doing in my PhD with many collaborators, as you can see on this first slide. We know that humans have modified almost all ecosystems on Earth, and especially over the few recent centuries, intensification of land use and activities related to industry, urbanization, agriculture have led to an increase in a change in biogeochemical cycles, and an increase in nutrients that are key elements in driving biological community diversity and dynamics in every ecosystems. In addition, increase in CO2 levels have changed the climate and enhanced greenhouse effect. So increasing temperatures as well as the change in nutrient availability has had great impacts on biodiversity and has even led to extinctions of whole populations and changes in the biogeography of species. So in lakes, we saw that the main impact of human activities over the recent centuries was the increase in nutrient inputs, which causes eutrophication. And you can see an example here of a large cyanobacterial bloom in Lake Erie in 2011 that caused major problems for drinking water for the whole population in the region, and it's also impairing ecosystem services that human derives from lake ecosystems. So we know that cyanobacterial abundance is favored under warmer climate and more nutrients. And we also know that, I just said that, there's a loss of ecosystem services, but also ecologically it's a very significant impact on the whole food web. So phytoplankton is the main source of food for zooplankton, which is the source of food for fish. And by causing hypoxic conditions sometimes, or by producing toxins, cyanobacteria will directly or indirectly break the whole food web. So we know that abundance of cyanobacteria is controlled by climate and nutrients, but what we don't really know is how these alterations of ecosystems have changed biodiversity of cyanobacteria. And as I said, this might have major impacts because some cyanobacteria accumulate and form large blooms, others don't. Some produce toxins, some don't. So it is very important in a lake management perspective. So the aim of my project was basically to reconstruct temporal and spatial patterns in cyanobacterial diversity. And I was mostly interested in knowing what are the factors, the regional factors, such as increase in nutrients, and the more, the local factors, or the more regional or global factors like change in climate, in influencing the composition, the biodiversity, and the biogeography of cyanobacteria. So to investigate that over several decades, I used an environmental DNA approach on lake sediments. And I don't want to explain too much about the methods, but briefly we collect sediment cores in several lakes. And as you can see here, we have around the Alps, the region I studied. Don't know how that works. We have mostly lakes that are varved, as it was explained very well before. So this makes it very easy to date the sediments, and we can have a very high resolution, an annual resolution in the dating. Then we can collect sediment samples at different depths that we are interested in and directly extract DNA from those samples in the clean lab facility. From there, we can put these samples in a sequencer and I targeted the 16SR DNA amplicons for cyanobacteria specifically. So it's the so-called metabarcoding approach. And after filtering and bioinformatics cleaning of the sequences, I used phylogenetic analysis to determine the species composition, but also the structure, phylogenetic structure of the communities. As I said, I did that in a few lakes, 10 lakes located around the Alps, so they're not alpine lakes, but all around the Alps. And this region has been very well studied, and this is great because we have high resolution data on nutrient concentration in the water column over several decades, and we have information about water temperature in the water column at different depths. So this is just an example of one phosphorus curve. As it was explained before, many lakes worldwide went through a phase of eutrophication in the 20th century. And in most cases, or in some cases, at least in Europe, efforts were made into restoration of lakes, and that led to a decrease in phosphorus concentration. So I used these nutrient concentrations, temperature, and a bunch of data to try and understand what are the main factors explaining diversity and richness of cyanobacteria over time in those 10 lakes that are listed there. And what I found was that temperature had the most significant impact on the number of species of cyanobacteria that were found over time. And as you can see, the relationship is quite strong, and it's significant as well, statistically. So here you can see on the y-axis the number of species found in the sediments at a given year. Most of these samples are taken with a yearly resolution. And on the x-axis, you have the normalized annual maximum stability index. So what I forgot to say earlier is that temperature has it can have a direct impact on the metabolism of organisms, but it also has indirect impacts by changing the stratification of lakes, intensifying the stratification, or changing how the nutrients can be recirculated during the year. So higher thermal stratification led to more species of cyanobacteria, basically. So we look into more detail to know, well, which species are favored. Are there species that pop up and are more favored by these conditions? And what we found was really interesting. You see on the left side, so these are main groups of cyanobacteria. And on the left side, you have the three groups that potentially can form blooms, and they can also be toxic cyanobacteria. So all of those were, the richness, so the number of species of those groups were particularly favored under high temperature and under the recent conditions in those lakes. As I mentioned before, we used phylogenetic analysis to look more closely at how cyanobacterial communities changed over time, but also how they compared between all the lakes of our region. And we identified, this is just a cartoon phylogeny to help explain this. And what we observed first was that there was an increase in the prevalence of those bloom-forming cyanobacteria that I mentioned before. In the early 20th century, they were not found in many lakes, and then over time, they sort of appeared in most of our lakes across that region. And then we also found out that these species of bloom-forming cyanobacteria, not only were they more prevalent, but it was also more of the same species that was distributed in all the lakes. So I will walk you through this plot that shows community similarity on the y-axis. Higher number means communities are more similar. And the x-axis is time. So I grouped all my lakes per decade, and then I compared the diversity in all the lakes to one another. So it's a pairwise comparison of the phylogenetic diversity. And the violin plots show the distribution of these pairwise comparisons. So you can clearly see that there was an increase, especially over the last few decades, in similarity across all of the lake communities. And this was caused, of course, by the spread of certain bloom-forming cyanobacteria like Dolichospermum lemmermanni, for those who know a little bit, or Planctotrix rubescens, which are famous harmful cyanobacteria that can form blooms. So just to a quick sum up of my main or key message, I think that lake sediments provide really crucial information on past community dynamics, not only composition, but how these cyanobacteria behave or change in a rapidly changing environment. And also sedimentary DNA is very useful to reconstruct the past diversity of cyanobacteria and other organisms that don't leave fossil records. So we can really go and extract the DNA from the sediments and target whatever organism we are interested in. And we saw particularly that there was an increase in the prevalence of potentially harmful cyanobacteria across this region. It would be interesting to sample more lakes and find out if globally there is also such a change. And this, in our case, this was most likely favored by warming and environmental changes. So all the lakes I study are not limited by phosphorus or nitrogen, except maybe one, which is on the lower end. But this huge pulse of nutrient addition to the lakes probably favored a reset, or it gave the chance to several taxa, in my opinion, to thrive. And the conditions that are now very homogeneous across all those lakes probably really made it possible for some taxa to disperse. So overall, yeah, there was a homogenization of the cyanobacterial communities. This is extremely important in terms of lake management. So just reducing phosphorus was not enough, or didn't really, it helped in reducing the abundance of cyanobacteria, but it didn't really help in terms of the community diversity. Now we're stuck with potentially harmful cyanobacteria more than before, actually, in some cases. And I just want to mention briefly that, so now we did this over what limnologists call long-term. Of course, it's relatively short-term, and we're now working on using this method to go back a little bit further back in time and try to investigate not only cyanobacterial diversity, but the diversity of the whole food web, basically, by using a combination of more classical markers and sedimentary DNA methods. And with that, I would like to thank my collaborators and a bunch of people who made this project possible, as well as the funding agency, Swiss National Science Foundation. Thank you very much for your attention.
What is the effect of the diagenesis on the preservation of your DNA? So, and the other question that follows up is how far back in time can you go without having a bias due to diagenesis?
Yeah, that's a very good question. That's why we started with relatively short term, because we had monitoring data as well on phytoplankton, and we could really compare over at least 8 decades or so. And with historical records, we also know in those certain lakes, we know, for example, when Planktotrix rubescens, that red cyanobacteria there, we know when it appeared, or at least when it was first recorded, and our data really corresponds well with those records. But of course, we have to test the quality of the DNA and verify that there are no obvious patterns of preferential degradation over time, which we're very careful with. Stringent filtering with the bioinformatics steps, and yeah, for this new study, we also have to reduce the size of the amplicon. So it's a trade-off. To do phylogenetic analysis, it's better to have the longest amplicon possible. But to go further back in time, I think it's important to reduce a bit the size. But I think, yeah, other people have shown in the ADNA session that it's really possible over much, much longer time scales, and it's really promising. Yeah.
I'm going to present to you the preliminary results of my postdoc that I am carrying out in Toulouse, also with people from Besançon and Orléans. I'm very happy to be here because having some difficulties in interpreting some of the results, and I think you people can really help me out, because I really need to renew my contract in Toulouse. So we know that mountain lakes are extreme environments, very sensitive to global changes, and also, as we have seen in previous talks, also sensitive to local environmental forcing, including anthropogenic influences in lake catchment. Mountain lakes are also remote localities, difficult to reach for most organisms, and this is also relevant in this presentation. One example of local disturbance is the use of catchments for livestock grazing, which increases the nutrient loading into the lake. Okay, but today I'm going to talk about the fish introduction in previously fishless lakes, which is the case of most lakes in mountain areas around the world. because mountain lakes are typically separated from lowland areas by, for example, large waterfalls that are very difficult to overcome by fish in this case. So in the French Pyrenees, the fish introductions began at the beginning of the 19th century. There's a big increase in the number of lakes stocked during the 60s thanks to the use of the helicopter. So we have now 90% of lakes in the Pyrenees stocked with fish. So it's a big real problem now. Most of the species introduced are of the family of Salmonidae, including the trout and other species coming from other parts of Europe and even North America. And we also have this little fish here, Phoxinus, which is not of the same family. This little fish is used by fishermen to as a bait to catch the trout, you know. And at the end of the day, this little fish would be typically released into the lake by fishermen, causing also big problems. So the effects of fish can be indirect, as demonstrated by this paper of these authors, who studied 3 lakes in Sierra Nevada, California. They used these 2 lakes where they know that the trout were introduced in 1965, and they measured the pigment content in the lake sediment. So we can see an increase of the pigment content. In one of the lakes, the trout went extinct because it was unable to reproduce, so the pigment content in the lake sediments went down again, and this lake as a control was used here. So this is because the fish alters the nutrient cycles by predating on invertebrates that live in the benthic areas, and after the consumption, with the excretion of these nutrients, they make available these nutrients to the plankton. And so it happens also when the fish are predating on terrestrial insects that fall to the water surface. They accelerate the nutrient transfer from land to the lake. The direct effects of fish are based on the fact that fish like to predate first on larger prey, as we demonstrated in this paper, where this species is not only the largest one, but also the one who likes the most diving into the water column, making it more vulnerable to fish predation. So this is why the amphibians are typically the first organism to disappear after the fish are introduced. And this little Phoxinus is also causing big problems, which is released into the lake by fishermen, because he likes a lot eating on the Daphnia, which also control for phytoplankton. And this is in another project led by Dr. Mark Ventura, which they demonstrated that after the Phoxinus removal, the water transparency, the water became clear again, because the Daphnia were again able to control for the phytoplankton blooms. So in the study, we hypothesized that the Daphnia size could be also subjected to changes. The Daphnia would be able to reduce their size in order to reduce the risk of being predated by fish. We actually do not measure the Daphnia in the sediment cores, because these are not really well preserved, but the Phipia, which are the eggs laid by the Daphnia. But there's a very good relationship between the female size and the Phippium size. So we can use this Phipia in lake sediments to know a bit about the size of the Daphnia. And also we use the chironomids, which are very interesting because they have a larvae with a head capsule that is made out of chitin, which preserves very well in lake sediments, so we can recognize the species also. So the chironomids are also interesting because some of them also contain hemoglobin, which is particularly is an invertebrate hemoglobin, which is very efficient in trapping oxygen and at very low oxygen conditions. And so this is advantageous in front of other chironomids when there is a large quantity of organic matter. So for the trip chironomini and the subfamily Tanipodini, which are basically the most, the two groups of chironomids that contain this hemoglobin, we can expect a positive indirect effect of fish due to this alteration of nutrient cycling, which is which makes the lakes a bit more richer in organic matter, which will be the same for the Tanipodini, but in the Tanipodini there is a particular thing that is the pupa needs to go up to the surface of the lake to breathe. And that makes it more vulnerable to fish predation, to the direct effect of fish, which in this case would be negative. So the hypothesis in this case would be that the chironomid assemblages are affected by fish presence because not all chironomids are affected the same way. And then the aim of study would be, in this case, inferring chironomid communities in historical sediment records. So we visited some lakes. We took sediment from the deepest part of the lake in several lakes, and we looked for the Daphnia and the chironomid communities. So I'm going to present preliminary results for 4 of these lakes very quickly. These lakes are located in the French side of the Pyrenees, in the eastern part, are located in granitic bedrocks, which make them to be slightly acidic. They are quite small, particularly Lake Romareda. And these lakes have been introduced with trout, and these other 2 lakes also with Phoxinus. So in Lake Siriu, well, we're not going to get into the details of these chironomid species that we found in the first centimeters of the sediment core, but we can group all these species in these 2 chironomid groups I talked to you about. Okay, so we can see that after fish introduction, the chironomini were increasing, and the Tanipodini maintains stable, possibly because positive and negative effect were maybe counteracting each other. As to the presence of fish and the presence of livestock grazing in the catchment, we can see that these results are visually, at least, they are easier to relate to the presence of salmonids. And as to the size of the amphibia, we measure the length, and we can see that after the fish introduction, the length of the amphibia reduced in size after some delay in time, okay? From more or less 1 millimeter to 0.7 or something. In the second lake, also many chironomid species, which we can group again. We can observe also increase in chironomid. This time we also observe a reduced abundance of Tanipodini, making it clear that the negative effects might be stronger than the positive effects. And again, these results can be related better to the presence of salmonid. But then, and this is the Phoxinus also, which was introduced, we don't know when, but possibly around the 80s or 90s. So we lack some information here. But the problem is that the Daphnia here apparently changes the size before the fish were introduced. So we may ask why this is so. I am rather interested in why this is happening to me, you know. But in any case, we can think about a different hypothesis. For example, that the Daphnia species changed, so it's not really the same species, because we can observe here quite large change in the amphibia length. So this might be for reasons other than fish introduction. In the third lake, we can do the same, and the chironominis start to increase with some delay, and the Tanipodini apparently increase and then decrease again. We don't know why. In fact, in this lake, fish was already present from a long time ago, but was very scarce, probably because it's unable to reproduce. But at the 1950s, the fish was reintroduced intensively in this lake, so we might expect some changes here due to this fish reintroduction. In this case, well, this is the figure again with the number of fish stocked in this lake, and again this figure is the same for all the lakes, okay, because there are mean values for all the valley where all the 4 lakes are located. And in this case, well, this is the Fuxinus, again, somewhere here. And the Daphnia, in this case, did not change really the size, but the size of the Daphnia are quite low in this case. So maybe they changed the size before. So we don't know. Finally, the last lake, La Gardelle, we still don't have the datation, but we think the fish introduction was probably around the mid part of the graphic. We don't really observe many significant changes or very clear changes here, so it is difficult to know. Well, this lake we don't have any information here, but the Daphnia really didn't change the size either. Okay. So the preliminary conclusions here would be that the introduced fish in mountain lakes has a potential to change chironomid communities, either by favoring indirectly the chironomini and taniporini, that would be a positive effect, or predating directly and preferentially on the taniporini, that would be a negative effect. Also, that the effect of fish on chironomids appears to be stronger than that of livestock grazing. And finally, that the fish presence is perhaps related to the reduction in Daphnia size. This appears to be the case in one of the lakes studied, but at the moment it remains unclear for the other lakes. Okay. So thanks for your attention.
Have you looked at Kyambogo's phantom midges?
The what? Sorry?
Kyambogo's phantom midges?
Oh yeah, in one of the lakes we found, only in one of the lakes we found the mandibles of the Kyambogo. But it was in the last lake, but there's no changes. We found not too many, but constantly along the sediment core.
And it's the same species throughout?
Of the Kyambogo, I don't know. I don't know. I know it's Kyambogo, but I don't know the species.
Okay. Well, secondly, also the Daphnia. Typically, you can also identify 2 species.
Yeah.
The Amphibia.
I am getting some training also on that. But I didn't have really the time to perform this distinction before this meeting. But I think in that lake where the change in size occurs before the fish was introduced, I'm sure that there is more than one species. So we really need to separate this.
Okay. And then finally, are you aware that perhaps that also during the fish stocking itself that new species of Daphnia can be introduced?
Well, I didn't think about it, but maybe it is so. It is interesting to have it in mind. Yeah, I don't know.
About the trophic state of these lakes, you have any data, pigments or whatever, that show an increase in the trophic state after the introduction of the fish? Or because you mentioned it's a cycling will change and all. Yeah.
We are carrying out the analysis, and right now we don't have the results. Dr. Teresa Buchaca is doing some pigment analysis, and about the trophic state, we still need to analyze the water samples. But I think these lakes still remain on the oligotrophic. I'm not sure, but I think they are also quite small, so they could be also mesotrophic. But definitely it's in the thing to look at, the sediment, the pigment content change, for example.
Hello, my name is Rosalie Bruel. I'm working on lake response to climate change, and especially on lakes that are heavily impacted by human activity. So for the people that are not that don't have an ecological background, maybe they will think that when there's a linear response between the forcing and so the ecological response, so that means that when the forcing increases, you get a response, but if this forcing decreases, then you go back to the previous state. But for many people in this room, they already know that it's not always true. There is some nonlinear trajectory. One of this nonlinear trajectory is regime shifts. Regime shifts are triggers large, abrupt, and persistent change in the function and structure of the system. Regime shift can take place have been reported in many ecosystems worldwide, and the driver can be both local or global. The mechanism beyond regime shift is depicted here on this graph. So the idea is that initially a system is in the stable state, so the state A, and even if you have some drivers that act on the system, the ecosystem will remain stable and resilient in front of this forcing. But as the driver increases, so as the pressure increases, the ecosystem loses resilience. So the ecosystem is pictured with a ball here in this landscape. And once it crosses a threshold, the ecosystem falls into an alternative stable state, state B, that is stable as well. So the implication for management is that even if you decrease the driver, the ecosystem will remain in this stable state. So this regime shift has an important explanatory power. So it has been many transitions have been characterized as being a regime shift because the restoration measures have no effects and cannot guarantee the return to the previous state. But some reviews are suspecting that many of these so-called regime shifts are in fact not following this kind of trajectory, because they are not, they did not quantify the actual mechanism. So, in fact, what is needed to quantify this kind of relationship is a three-step way. So first, you, of course, need to identify the transition, the abrupt transition, because it's the first parameters. Then you need to identify the driver of the transition. And finally, you need to identify the mechanism of a regime shift. So I already talked about the B stability, the fact that a system can be in two states for the same level of drivers, so state A and state B. But there are also other mechanisms, such as loss of resilience. So as an ecosystem is approaching the threshold for the shift, its variance and its autocorrelation of the monitored ecosystem state proxy will increase before being low again once it reached the alternative stable state. So what you need to do this quantitatively operational assessment is a different thing. You need to have long-term data that are sufficiently high resolution to characterize ecological processes. You need to have drivers and responses, and you need, of course, to use some statistical tools that can guarantee the reproducibility of this method. So we wanted to know if we could detect regime shift trajectory in lakes. We worked on Lake Jaurès because we thought it was a good system to do so. It's a lake that has been heavily anthropized and that went under hypertrophication in the 50s. And despite the restoration measure, we do not observe the return to the previous condition. But the thing is that since the 80s, since the 1980s, the air temperature has increased even more in the Alpine area. So in fact, this apparent stability could also be the result of the combination of new factors, because this is also a really important possibility that has been evaluated. And also because Lake Jaurès is much deeper than a system where regime shifts usually take place, there is just no feedback mechanism that could explain this kind of trajectory in that kind of lake. Our alternative hypothesis is that indeed this absence of return is the consequences of a regime shift in this lake. So in order to fulfill the requirement we identified, we worked on a core. So it's a short sequence. It's covered 170 years. If you want some detail about the dating, we can talk about that afterwards. But we reconstructed with biological proxy responses through cladocera and drivers through, so mainly total phosphorus, because as it has been pointing out this morning, it's a main driver in lakes, through diatoms and pigments. And then we used the climate database for the past, covering the past 200 years. So then we combined different statistical tools to assess and to answer the step I mentioned before. So this includes ordination methods in order to summarize the ecological information. Then we detected change point in this. We assess the contribution of driver with general additive model. It's really relevant for this kind of records because their strength is that they allow different contribution along time of the driver, which is closer to the reality. Then we looked at the actual regime shift mechanism, which is the response versus forcing relationship, and if we could detect any loss of resilience. So these are the results. First, this is our ecological response in Lake Varese. So we summed, as I just say, we summed up this information into ordination axis, and what we see is three transitions. The first one in 1926 corresponds to a slow, like a linear increase of Daphnia species. The second one is abrupt and in 1946 and corresponds to a total reorganization of mainly the pelagic habitat. And finally, since 1988, there is a new assemblage within Lake Varese. Because so all those species, all those cladoceran species are pelagic species. They are really represented in our sequence because we took a deep core. But we wanted to look at also at the littoral taxa. So we carried out the analysis only on the littoral species. So here are the results. We found two transitions, one in 1946 and one in 1988. So after that, after we detected the change point, we looked for the drivers. The drivers that were identified were the mean air annual temperature. So as you can see, there is a trend you're probably famous with, the increasing trend over the last century with +2 degrees in the area, so it's twice the global average. And so the phosphorus, as suspected, was also selected. There was a really important increase in Lake Varese. I told you about this hypereutrophication because the level went from 30 micrograms per liter to more than 200 micrograms per liter from 1948. So the general additive model selected both forcing to explain the variability in the total assemblage and only phosphorus for the littoral assemblage. Overall, the explained variance was pretty high, so we were pretty confident in the strength of the model. So now let's have a look at the trajectory. So it's response of the system plotted against the main driver that we identified, so it was phosphorus. And chronologically, this is a relationship. So you have the initial, what we will call initial in our study states. From 1926, there is a response of the system to a slow eutrophication process. After 1948, there is a total restructuration of the system following the sharp increase in total phosphorus. And in the last decades, there is a new state that have been observed. If we do the same thing in the littoral, so there was the initial state before 1944, and from 1946, so for a slow increase in driver, we fell into another state. So what we see here follow and the recent, and of course, yeah, the recent trajectory following the restoration, the ongoing restoration. What we see here follow for both total and littoral habitats what we expect from a non-linear relationship. There is this loop, it's not taking the same pathway than when it shifted. But we need to account for the information that we got from previous analysis, which is that for the total assemblage, annual air temperature is really important and a really important driver. So we know that because here when you look at the general additive model, there is a really high contribution of air temperature from for the last 30 years. And when you look exactly at the relationship, so between the contribution of temperature to the assemblage versus the actual temperature, you can see that it's a monotonic relationship. So it's easy to model. We were able to subtract it from the total response in order to get only the nutrient response. So again, so this is the scores for the total assemblage without the variability explained by temperature, and this time we do not see the stability again. So finally, the last mechanism for regime shift we looked for was again the loss of any loss of resilience through the variability in state variables. So here I present you autocorrelation. So here there is the increase before the break is significant, but we do not see a real stabilization. Overall, it's increasing along all the sequence, while in the littoral habitat we have a significant increase before the transition, followed by a decrease in autocorrelation. So this follows the expected signal. To sum up, despite having an abrupt transition in both total and littoral habitats, and at first some B-stability in both assemblage, once accounted for all the drivers identified, we could not detect any regime shift trajectory in the total assemblage, but instead a combination of new forcings, while in the littoral habitat, all what we were looking for were verified, and what we observed is true nutrient-driven regime shift, but at the scale of the littoral habitats. And the return of the species here are in fact ubiquitous species. So while we had some macrophyte-associated species before, we now have some species that just that would deal with any condition, so not as exigent. So the key message here is that with this reproducible workflow analysis, it would be possible from high-resolution paleo data to reconstruct the actual frequency of regime shifts in lakes. Also, from a management point of view, it's important to consider the different habitats to evaluate the state and health of the ecosystem, because here we saw that littoral communities were pretty good sentinels. And finally, again, once more, it's proven that the combination of local human stressor and climate change is a really important parameter to keep in mind. So thank you for your attention, and this is my the people who worked with me on that project, and also the workflow analysis follows IDs from the Pages Aquatic Transition group. So thanks to them as well.
You're using the cladosaur to say there's a regime shift. Did you compare with other, community like a diatom? Are they shift at the same time, or? You mentioned the shift is a state shift or only the community shift in the lake ecosystem?
Yeah, you mean if I do the same analysis with diatoms, for example?
Yeah, do you think they are responding at the same time? Or?
So we use diatoms to reconstruct phosphorus. So we didn't want to use them as well as an ecological proxy, and in fact we had to combine diatoms with pigments in order to get an accurate reconstruction of phosphorus, because diatoms seems to have a delayed response. So they respond really rapidly, but a bit delayed. They do not depict the early eutrophication stage. So in that prospect, we rather work on Cladocera because they're more sensitive to this bottom-up effect. I don't know if that's answers.
Thank you.
So I'm going to take you to tropical Southeast Asia, quite a bit different from what we've been hearing so far. And because this is a session on human impacts on lake ecosystems or aquatic ecosystems, I figured I'd also try and give a very short overview, as best as I can in 2 slides, of what these human impacts could have been in this area, in Malaysia. So I think I have to sort of take our story back to around about the mid-19th century, when during British rule, mining started to really be developed in Malaysia, still on a localized spatial scale. Similarly, plantations started, notably rubber plantations at first, and it all started in the west where the bigger settlements were, and slowly, but also the developments moved eastwards across the peninsula. This was sort of enhanced, I would say, in around about the mid-20th century when the Malaysian government came to place and initialized a development plan called Velda, where they actively relocated farmers, created new rural communities to stimulate things like rubber plantation and oil palm plantation. And this is sort of, the Velda project has stopped by now, but it has sort of continued on to this day, that this sort of increased intensity of agricultural activities. And by now, for people who have visited the peninsula of Malaysia, if you drive east from Kuala Lumpur, basically what you'll see is oil palm after oil palm after oil palm. So dramatic changes in the landscape across the last decades to a century, I would say. And then on top of that, towards the bottom right, you also see that there's mining activities, in various places in the country where people are mostly digging up bauxite, so aluminum ores or other metals that they are looking for. To my surprise, because I didn't know this at first, but there's not a lot of freshwater ecosystems in Malaysia. Actually, naturally speaking, there's only 2. There's a site called Tasik Bera, and there's a site called Tasik Chini, which is the one we are working at, and for which I will show you some results. Both of those sites are situated slightly to the east of the peninsula, I would say, and Tasik Chini, which some of you might be able to see on that tiny picture I put on the screen, is a flood pulse wetland. I've been sort of describing it as a sort of drowned river valley. It's quite complex. It's not a nice little circular lake. It's actually 12 basins that are all interconnected with various inflows that are depending on monsoonal seasonality. And then to make the situation even more complex, some people might be able to see, well, pointer's not working, but a very small dotted line in the northwest of the wetland, which is an outflow where lake water will drain into the River Pahang, the biggest river in Peninsular Malaysia, during dry season. But then during monsoonal season, the situation is reversed and the Pahang River can actually drain into the lake. So it's all quite complex as to how this ecosystem has been functioning in terms of its hydrology. But it's an important ecosystem because there's only 2 freshwater sites. There are hotspots for regional biodiversity. Lake Chini actually has a few endemic fish species occurring as well. Plus, there's still a community of about 400 native inhabitants of the Orang Asli tribe that live around the lake and that use the lake as a primary source of food, of drinking water, etc. So, for their livelihoods. Now, having said that, sort of on top of these regional or national developments that I've been describing so far, there's also a couple of local developments. And one of the most important ones was the construction of this dam in 1995. This dam is located in that inflow-outflow river that I just described to you. And it was basically constructed to ensure that ecotourism would not suffer during periods of drought or during periods of high water. You can nowadays visit the lake and you can take a nice boat tour around these 12 basins and you'll see beautiful lotus flowers, etc. And that works quite well. But what people were finding that towards the end of the 1990s or 1980s, beginning 1990s, lake levels were falling ever further and that was hampering this ecotourism. So to ensure that it had stable lake levels, they built this dam, but then obviously that had all kinds of other consequences as well. And one of them was that the fish exchange from the River Pahang into the lake obviously was dramatically cut short. It was still happening, but only during very major floods, but not sort of regularly during the year anymore. Other effects were noted as well, especially by people from the local community, such as changes in the aquatic macrophyte composition, etc. And then one final driver of sort of what we expected to affect our lake ecosystem is this beautiful view you have. If you take one of these touristic boat tours and you're on the lake, you see this massive, massive mine right on the border of part of this wetland ecosystem as well. And this mine has been active since, we think, mid-20th century, on and off again, and it's obviously been increasing in size as well. It's currently deactivated, but you can still see orange flush coming in as soon as it starts raining. That basically washes straight into the lake. So obviously, we thought that might have an impact on our ecosystem as well. Because if you listen to the local people who live there, they will claim that there's been quite a few problems recently, which they sort of summarize as ecological deterioration. So the fish stock has declined, so there's fewer fish, they're smaller, and also the diversity of this fish pool actually has declined as well, according to the local tribes people who use them as their main food source. Similarly, lotus flower has been disappearing from the catchment, been replaced by one of these invasive macrophytes, and according to the local people, water quality has deteriorated as well. So what we try to do, similar to previous studies we've seen in this session, is use our paleolimnological toolkit to get a picture of, well, spatiotemporal changes in this ecosystem, see if we can actually identify which culprit did it, if you will, see to what extent the effects now are different from natural variability in this ecosystem, and hopefully in the end, and we're definitely not there yet as this is work in progress, maybe do some recommendation as to how this wetland can actually be saved from going completely to waste, if you will. So as other people here in the room probably also have done, we collected gravity cores from all around the lake ecosystem, focusing on all these different basins that we have. For 3 locations, we also took long piston cores, long in this case being 2 or 3 meters, so it's not really long, but long enough for us to get a good picture of, well, let's say, millennial scale developments. And for these 3 sites, we tried to choose sites that were different. So the site in the southeast, that's marked there with the red asterisks, is close to the mine, so we've called it the mine site, whereas the one on the west, for us, was the most pristine part of the wetland, still being surrounded by natural-looking forests, whereas on the east side, for instance, you have all these plantations. So we tried to have a contrast between what we thought would be most heavily polluted towards what we thought would still be most pristine kind of situation. And that's the 2 or 3 sites I'll be focusing on for today's talk as well. Just as sort of, I'm not going to go into the details here, but we're also carrying out a monthly monitoring program at the moment, so we're collecting data on changes in water chemistry with the seasons, we're collecting sediment trap data, etc., just to find out more about how this ecosystem actually functions throughout annual cycles, because not a lot is known, unfortunately, to this date. So some results. Basically, I actually wanted to start with focusing in on the chronology itself, because there's normally a tool for us to sort of see how old our sediments are, but I think for us it's actually also one of our major results, at least to this day. So the first thing we did is we tried to lat-210 date the upper parts, or the most recent parts of our sediment column, and what we see is a more or less steady decline of lat-210 activity with depth, so it's rather easy to plot an age-depth model using these data. But that lowermost stippled line that you see there is the sedimentation rate that's derived from this lat-210 profile. And you can see that around about 1950, so towards the right-hand side of the plot, you see those last 2 points are really much lower on the sedimentation rate axis, which is the right-hand y-axis. So that, for us, showed basically an increase in sedimentation rate at around about 1950. But unfortunately, it's based on these last 2 lat-210 dates, which have huge error estimates associated with them. Luckily, just before this week, last Thursday, I got the results in for some radiocarbon dates that we also did on the older parts of the sediment sequence. And what we see there is, first of all, our wetland came into being approximately 6,000 years ago. That does make sense when we look at basal dates from coastal swamp areas, where also about 6,000 years ago they see the onset of sedimentation there. So it seems to fit a more regional picture of regional sea level rise 6,000 years ago. We also see relatively stable sedimentation rate, especially if we translate that to centimeters per year, or millimeters per year in this case. And finally, we see that definitely this upper part, these last 70 years or so, since 1950, show about a 5-fold increase in sedimentation rate, which is a huge change compared to the natural or previous sedimentation rates of around about 1 millimeter per year. that obviously then sort of, if you see such a thing, the first question then ask is, okay, so what is that? What is that extra sediment? Where is that coming from? What is that composed of? So quickly going to one of my favorite proxies, because it's simple, it's robust, and it's easy to apply. Loss and ignition. We applied that to all the sediment records we had, and I'm showing you the 3 that I introduced to you earlier today. And we see relatively similar profiles throughout the entire wetland. But we also see at around about 1950, when our chronology indicates that we have that increase in sedimentation rate, we see a dip in the loss and ignition, meaning that we have relatively fewer organic material, or basically more clastic material from that point. Second question then is, what kind of clastic material is that? What is that composed of? Did something really change? We try to address that question by applying a number of different methods. You see here XRF counts. We also have ICP-MS data and XRD data. And we're still sort of trying to figure out really what's happening throughout the entire wetland. But if we look at this site that we're just focusing on with our chronology, and we look at 1950, then we can basically look at these figures and say, well, not a lot seems to be changing here. So this for me indicates that, yes, we've got more sediments coming into our lake, but the sediment source didn't really change. It's not like all of a sudden we got atmospheric pollution coming into our lake, or all of a sudden we got flash floods bringing in sediments from different parts of the country as we had before. So for me this says more material coming in, and most likely we're thinking this is soil material, obviously from directly around the lake, that's increasingly coming into our site from 1950 onwards. We see this for the largest part of the wetland. I would say the situation is slightly different when we look at that one record we have that's close to the mine location, where we do see more variability, and especially around 1950 we see a decrease in the component of zinc, a little bit in potassium oxide. We're currently working with John Boyle in Liverpool to try and make sense of what this exactly reflects, but it does seem to suggest that slightly different type of material that was washing in here. And we're now asking the question, can that have something to do with initial mining activities perhaps already starting around about this time? Maybe that different parent material would be available for influx. Ooh, Natalie has accelerated time since I started to stand here. Okay, I'll speed up a little bit then, sorry about this. Because then the last question obviously is, if we get more influx of material, you would expect that to influence the ecosystem itself as well. And we've tried to analyze that by looking at the algal material. So diatoms, and Keely has been instrumental in developing this record with a student of ours called Charlie Bridden. We've complemented the diatom analyses with pigment analyses, which tell us something about the algal flora as a whole, I would say, and also includes information on cyanobacteria blooms. And to very quickly sort of highlight the things we think we see is that, first of all, we see already minor changes occurring way before the 20th century. This says eutrophication, that might be the wrong word to use here, but we definitely see changes occurring before the last few decades or the last few centuries that I've been describing you before. But we can also see that the major change does seem to happen around about 1950, where especially all the sedimentary pigments increased dramatically in concentrations, indicating that probably there was an increase in primary production around about that time. And in the diatom record, we see a shift within the diatom community itself, perhaps reflecting a lower relative abundance of planktonic taxa. Finally, we do see changes towards the top as well, around about 1990. That's just visible in this one record that's near the mine. Again, might have something to do with mining activity. And just as a last conclusion, and unfortunately I put the bar right across there, but we don't really see anything happening for this site at 1995 when this dam was constructed. Now, that might be due to the fact that this particular site is about as far away from the dam as you can get, or it might have something to do with the fact that the dam may not have had as big an influence as we've thought so far. We're currently applying the same kind of methodologies to the more pristine sites and the sites closer to the inflow-outflow as well, so hopefully we will be able to answer that question then. Last slide, because I see Natalie already holding up another little flag there for me to really stop talking. Just looking at the concentrations, the last 6,000 years basically had very low concentrations of pigments up to only about the 20th century, so we do think something really has changed, although this might also be partly influenced with preservation potential, so we're doing other types of analyses to back this up. But hopefully I've, in these 12 plus or minus 2 minutes, been able to convince you that we are working with a very unique tropical wetland here, where things are changing and where there is definitely indications for soil erosion that have dramatically increased sedimentation rates and have also influenced the ecosystem itself. So thanks to all the collaborators who made this work possible, and to you guys for being here.
Yes, I will be talking about integrated habitat and land cover change approach for Lake Victoria watershed in Eastern Africa. This is a method of, a mixed method where we use remote sensing, we use physical, chemical analysis, we use DNA in order to determine the impact of humans on aquatic systems. But I will also make reference to the impact of aquatic systems on humans, which made it possible for humans to evolve and eventually go out of Africa. Yeah, so that is the Lake Victoria Basin and the watershed that we are interested in. Most of this used to be covered by forests and wetlands, but it has now all been cleared because Kampala is here. This is the capital city of Uganda. My university is located between Kampala and Jinja. So we no longer have most of this vegetation cover. And that is mainly because of urbanization, industrialization, which is all associated with human activity. Yeah, and here we look at specific study areas. This is Nzoia, which is within Kenya. This is Nachivubo within Uganda. And then this is Simiyu within in Tanzania. This was a collaborative work that was done by researchers. So the different colors that you see will tell you how much vegetation still remains. And it's not much, of course. So this is a detailed analysis. Here we have Lake Victoria, part of Lake Victoria. And particularly this one, the blue color is a sign of how much vegetation has disappeared. So what you are having here is a reflection of what is here. And this is part of Nzoia, which is part of Kenya. Again, this is showing how much vegetation cover we have lost. So most of the rivers are quite naked. Yeah, so fish introduction is another impact that we have had from the humans. At the beginning of 20th century, Lake Victoria Basin had a multi-fishery industry where we had several fish species that were available to the community. But by the end of the 20th century, we had almost 3 species, including Nile tilapia, then Nile perch, and Rosalie argentea, when we had hundreds and hundreds of fishes that were being used as food. So that was really a very big disappointment. So these are some of the fish species that are now highly endangered because of overfishing, and the wetlands supported this fishery because some of the fish would need the wetlands for breeding. Now that they have been destroyed because of industrialization and agriculture, it's a very big impact on the aquatic resources. Yeah, the loss of cichlids that you all know, I'm sure everybody who knows something about Uganda would know the story of the introduction of Nile tilapia and how it has impacted the haplochromines and also the tilapines. At the beginning of the 20th century, we had 2 big tilapias, Oleochromis esculentus and Oleochromis variabilis, but because of overfishing, there were no more. And the introduction of the Nile perch made the situation worse and affected several species. Yeah, of course, we had the species decline, as I have already indicated. And the interesting thing now is, because of overfishing of the Nile perch, some of the species that were feared extinct are coming back. And that tells us that maybe we may have to have strategies that we can manage the fishing if we are going to keep the biodiversity. Some hope, at least. Then The major impact that we have had from the 1950s to the 60s was, of course, overfishing. And the introduction of the Nilo perch, the introduction of Tilapia zilai and Oleochromis niloticus was to make use of the resources of the big lake. We had a very big lake, but with very tiny fish species of the prochromines. So the fisheries department had a big issue. Do we keep this big lake with very tiny fish, or we introduce the Nilo perch? And of course has been recorded as the greatest mistake that has ever been done in the history of our world. So it has been a very big crisis. It is continuing, and a lot has to be done in saving biodiversity in that area. And I've already said that there is some hope that if the Nilo perch can be fished, not of course overfished, if it can be fished in such a way that it can allow other small fish also to survive in the same waters. Now, it is not fair for me only to talk about the Lake Victoria Basin. We have other emerging issues, also aquatic systems, especially the Alpine region. With the discovery of oil and gas, this is another water system that is going to be impacted. This, sorry, I moved this so fast. So this is these are the rivers. This is the river that connects into the Mediterranean. This is the river that is coming from Lake Victoria. They are meeting here. What you are seeing here are wells, oil wells, that are being drilled in the middle of a national park. This is the hottest biodiversity area in Eastern Africa. Now we are drilling in for oil, and that is going to be another very big impact on our environment, especially aquatic resources. So this, as you can see, this is Albert. This is a network of national parks and reserves. And it is the same area where oil exploration is taking place. So certainly there is going to be a very big impact in this area. You can recognize this is George, one of the most productive lakes we have. This is Edward. So the green you see is the remaining vegetation cover. The yellow is the national park. The rest, this white is already degraded. And just next to Lake Albert, this is a grassland. The farming community that is here, their animals are dying because of, again, human impact. And of course with climate as part of that. So what we have here, this red is indicating the boundaries of the national parks. But as you can see, this area is a grassland. The temperatures are very high. The farmers, although they are next to Lake Albert, they have no water for their animals. So this is showing you how much degradation has taken place in the Albertine region. Yeah, these are satellite images. This is Albert. So this is a part of the forest. Bugoma forest is another area that has been much affected by human activity. The red portions you see, it shows how much the forest cover is being degraded. So again here, all this is a level of degradation. Forest cover change in Budongo. Budongo is one of the most studied forests almost in Africa. And because of our British connection, most of the good timber you have in Europe, its origin is from Budongo. The Movule and the mahogany all come from here. So this is George, this is Edward. Again, you can see all this is bare, and it's going to be a very big impact on the water system here. So we only have an island of the forest cover, but the greater part of it is already gone. Yeah, this is Bwindi. Bwindi is one of the parks in Uganda that gives the income that we do have. But again, here you see the red is telling you degradation is already on the increase. So how do we do? As far as the national parks are concerned, if these parks are going to survive, we must have corridors that will allow this wildlife to move from one place to another. But if they are isolated in their national parks, we cannot save them. They cannot survive. And that would be a great loss. So these are the corridors that I'm talking about. All this is a network of national parks. If the parks are to survive, we must have the corridors where the animals can move at different seasons. Of course, this is Lake Albert, as you can see. And again, you see not so much forest cover remains. So these are the corridors that we have talked about. This is Lake George, Edward, but all around it's already bare. Of course, this is our Great Lake. This is Victoria. When I was coming the other time with my colleague, we did not see from Madrid to Zaragoza, we did not see a single river. We did not see a single lake. And when we looked back, what a contrast. In Uganda, you cannot walk drive more than 2 kilometers without seeing a river or seeing a lake. So the East African paleoclimate is extremely very important, and we're just referring to it. It is not only humans that have got the impact, but the climate has had. But what is important is the aquatic system we have in Africa has also impacted humans. It was the access to the aquatic resources that humans became more critical thinkers, more innovative, and eventually moved to the outside world. So I want to introduce you to these what are known as the amplifier lakes. These are the lakes that made it possible for humans to evolve wherever they evolved. In Uganda, we have an interesting area, Karamoja, which is opposite Turkana. It has a very big impact on the evolution of humans from Uganda. Then big lakes like Victoria has not had much effect on the evolution of humans. So I'm going to move very fast. Again, these are the deep waters that influenced human evolution. And since I have little time, I'll just move on. This is the interesting story that we can discuss. This Lake Chad is not an amplifier, not so much important in evolution. But lakes of deep color are very important as far as human evolution is concerned. There was, at times, people tend to think that we have always had waters, but this area, this period here, we did not have any lakes at all. The lakes are much later, and these lakes have influenced human evolution. So although we talk about humans impacting environment, the environment has also impacted humans. Now, those of you who do parametric analysis, you see, this is telling us the number of deep lakes. This is telling us I think this is East African. First of all, we start with the species. And we start with the change in the brain, how it is increasing. Then the number of species also increasing. The deep lakes also increasing. All these are associated and impacting each other. The same story here. Now, this is a story you have to understand if you have to explain how these water systems impacted humans and eventually moved out through here. This is now extreme desert, but this is friendly. Humans were able to move out at this particular time. Yeah, already there is evidence that the population is moved, and this is the genetics that we talk about. We are not going to go into details because there is no time, and I will just have to go to the conclusions. These are the threats to our forests. These are the different lists to our forests. All these are threats to our forests. Threats to our forests. Encroachment. Our conclusion. So the forests of the Albertine Rift and the vegetation, the Victoria watershed have been subjected to a variety of threats, as you have seen. And the amplifier lakes that I have showed you about have served as a natural barrier, and they might have contributed to the habitat fragmentation, reducing migration, expansion, dispersals, and of course increasing biodiversity. The East African amplifier lakes that I've talked about are a keystone in this scenario and may help to explain why East Africa became the place where our humans evolved. And of course, in the last 2 decades, we have been able to accumulate a lot of evidence to show that humans have impacted the environment, but the deep lakes of Africa have also impacted human evolution. Pages sponsored my participation, and CEDA and USAID supported these research efforts. I thank you for listening to me.